15th day of this 21-day FDP program. We are very fortunate to have you. And the group of participants, they are from the diverse field, veterinary sciences, agriculture, horticulture, and also extension specialist and staff members. So with this, very good morning to all of you, and once again, very warm welcome on behalf of the this course director and course co-coordinators and organizers. So I request to start your presentation, sir.
Okay, thank you very much, sir. So good morning to all, and we'll be having a lecture on recent advances and in molecular detection or other type of detections in plant pathogens and how we can diagnose the disease. And what were the earlier methods, how we were detecting the pathogens, and now what are the recent and emerging approaches by which we are detecting these pathogens, and how these methods are suitable and how these methods are effective. So we'll be discussing all those things. First, let me introduce myself. Myself, Dr. Sajjad, and I am working as a scientist in plant pathology at Central Institute of Temperate Horticulture, Srinagar. And mostly I'm dealing with these detection and diagnostic techniques. Most importantly, the viruses. But here I will be delivering for almost all the pathogens which we are encountering, whether it is a bacteria, fungus, or virus. So first of all, let me tell you, because this is a diverse group of people. So first, let me introduce what is actually the disease. So if we see the disease, this is a harmful deviation from normal physiological process. So any physiological process, sometimes when it gets disturbed, we can say this plant or this human or this animal is a disease. So any deviation. So when it disrupts the plant's normal structure, growth, function, and other activity. So we are calling it a disease. And this disease is a two-word combination. One is this and another is ease. When a person or a human or a plant or an animal, when it is away from the easiness, then we can say this human or this plant is disease. This means away, ease means from easiness. So when it is away from this easiness, we can say this plant or this human is a disease. So there are actually three things which are most important as far as the disease occurrence is concerned. So three things are important. One is a susceptible host. That host should be susceptible. It should not be resistant. That susceptible means that the disease should cause, the host should be susceptible, host should be able where disease can occur. Then we have a second most important is virulent pathogen. Virulent pathogen means the ability of the pathogen to cause the disease. When it is having the ability. Otherwise, if the pathogen is there, it is not having the ability to cause disease, then we can't say it will cause the disease. So it should be virulent. The pathogen should be virulent, but it is having the ability to cause the disease. And then third and most important is the environment. If the environment is not favorable, and even though host is susceptible and pathogen is virulent, but environment is not favorable, then disease will not occur. For example, I can say we have very important disease here, scab. Apple scab, it needs low temperature, it needs high humidity. In the earlier season, for example, in the month of March or in the month of April, it needs very humidity, high humidity, 90 to 95%, and low temperatures near about 15 to 20 degrees Celsius. But if this condition is not met at that time, the high humidity is not there and the temperatures are high, then this disease will not occur. Even though the host, for example, apple, it is a susceptible host, and Venturia inequalis, it is a virulent pathogen. So environment plays very important role for causing the disease. So now coming to the, when, for example, any plant, we are talking about the plant here, when we go to the field and we will see some kind of a stress in the plant. So we don't know whether it is abiotic, whether it is a biotic stress, which kind of a stress is this? Or sometimes we may be observing some kind of the these bodies of the fungus or bodies of these insects, or where those might be we can observe in the field. But sometimes we may not observe such kind of the bodies on the plant. So farmer or even scientist or even researcher sometimes gets confused that what actually it is this. For example, if we say the abiotic stresses, there will be no kind of a symptom Like there will be no this fungal growth, there will be no insect, but we will be confused at that time. Similarly, in case of viruses also, we couldn't see anything on the plant, but we'll see some kind of a symptom like mottling, we will see the chlorosis, we'll see the wilting. So we will be confused that what kind of pathogen or what kind of stress is this. So to avoid those things, we have a very important aspect that is called disease diagnosis and detection. So diagnostics is a very important step as far as the management of the disease is concerned. Unless and until we will not be able to find the cause of any stress, whether it is a biotic, whether it is a abiotic, whether it is a nutritional deficiency or anything. Unless and until we will not able to identify the cause, we will not able to manage that stress. So, for example, if I will take example of the viruses, some chlorosis will be there, wilting will be there, but same will be observed in, for example, if wilting is there, maybe it is a water deficiency. For example, if chlorosis is there or necrosis is there, maybe it is a kind of a nutritional disorder. So unless and until we will not able to find the cause, we will not go for the management. For example, if it is a viral symptom and we are going for the management through nutritional enrichment or through water enrichment, we will not able to cure that symptom, or we will not able to cure or manage that disease because it is a disease caused by virus. So this diagnosis or so this detection is very, very important as far as this management is concerned. So there are various steps for proper diagnosis of the disease. So first up, we should have a proper identification of the plant, whether it is an apple plant, whether it is a guava plant, whether it is a walnut plant. So first and foremost step is that this is a plant. For example, we'll take the example of this is an apple. When it comes to my mind, this is a plant, this is an apple plant, then I will recall the disease, what are the various diseases, what are the various deficiency symptoms, or what are the various insect pests which are attacking. So second will be the recognition of the healthy plant appearance. And third, identify characteristic symptoms. So these are the two things. First, we will see what are the characteristic symptoms in the diseased plant or stressed plant, and how healthy plant is behaving. So what is the difference? Then we will see the symptom variability, whether there is any kind of variability. For example, is there any chlorosis? Is there any necrosis? Is there any this mosaic type of a symptom? Or is there any other type of a powder type of a symptom? So we will identify the variability of the symptom. And then we will look for the sign of the disease, causal agent. What I was talking about, for example, some fungal bodies are there, some fungal mycelium is there, fungal mass is there. If it will be there, we can, to some extent, we can come to the conclusion, yes, this disease is caused by the fungus. Or insect is there, we will come to know, no, this is a stress which is caused by any pest or any insect. So these are the various steps which we have to follow when we will go for the disease detection or disease symptom, disease diagnosis. But afterwards, we will be taking the sample. If we will not judge what kind of a disease or what kind of the symptom they have produced, then we will take the sample and we will go for the pathogen detection. That is why we are frequently calling disease diagnosis. So we are diagnosing the disease, and then we are detecting the pathogen. And there are numerous approaches by which we can detect those pathogens. So you can see here that we need, for detection of those pathogens, we need reliable and sensitive detection methods. So there are 3S we can use for the detection. 3S means that our detection technique by which we are detecting the pathogen, we should have sensitive detection technique, that is a 1S. Then we should have speed, that it should be having the this rapidity. It should have the rapid detection. It should have the fast detection method. So speed or this sensitivity, and third important most is the specificity. So these three things are very important for any detection technique: speedity, specificity, and sensitivity. Unless and until these three parameters are there for any detection technique, our detection techniques will not be much reliable. So maybe whatever detection techniques we might be having available this time, maybe one among these three items, maybe two will be there, third will not be there. For example, if we will take the example of ELISA, enzyme linked immunosorbent assay. It is having a specificity, it is having the speedity, but it may not be as sensitive as another detection technique like PCR. So there may be one drawback in each and every technique, and then the second technique will come, and that technique will be having that merit in the form of some other aspect. So we need these reliable, sensitive, rapid, and robust diagnostic techniques or detection techniques for detecting these pathogens. So you can see there are numerous properties of any pathogen, or I can say the virus, or any pathogen I can say, by which we can go for the detection of these pathogens. For example, we'll be having when we'll see any kind of a sign, for example, powdery mass on the plant. We can see any spore through microscope. We can see any mycelium through microscope, and we can come to know that okay, this is a disease caused by some fungus. We'll not be able to trace which type of fungus and name of the fungus. Then we need different technique. For example, we will go for the molecular approach. And through molecular approach, we can identify the strain of that bacteria or fungus or virus, and we can identify the race of those bacteria. And there are various approaches by which we can identify those bacteria, those fungus, or those strains which are associated with these bacteria or these fungus. And you can see here number of approaches, and most importantly, we are using this polymerase chain reaction, then reverse transcriptase PCR, which mostly we use for the viruses, multiplex PCR, where we can detect more than one pathogen in one go, and then we have a real-time or quantitative PCR, where we can go for the more sensitivity of this PCR technique, or we can quantify the pathogen, how much pathogen is there. And then now recently those high throughput sequencing approaches, even if we are having the symptoms, but nothing is known and we are not able to check through PCR or other things, we have a technique these days that is called a high throughput sequencing, by which we can go for the sequencing and we can come to the know the cause of the disease. So importantly, that microscopy was very important in the earlier times, and nowadays is also going on for mostly for these plant pathogens or for these insect pests. And we use microscopes. First step to detect or to identify these pathogens or these pests or insects is the microscope. And we identify through microscope that this type of a spore is present, that this type of a mycelium is present. And then second and most important is the electron microscope, by which we can mostly identify or we can mostly detect the viral pathogen. Because these electron microscope is having the very high resolution power, and we can detect very minute particles of those viruses through electron microscope. So this is one way of detection of these pathogens. Then second is using detection using the ELISA, that is enzyme linked immunosorbent assay. That through these assays we can detect mostly viruses, or sometimes we can detect or we can identify the bacteria as well. Because this ELISA is totally based on the protein nature of the virus or any bacteria. For example, if we are taking any protein from the bacteria, and we will develop the antibody against that protein, and we can identify that bacteria as well through the ELISA. But mostly this ELISA is being used for the viral detection. And presently we are also doing this enzyme linked immunosorbent assay. And there are n number of these variants of this assay. But most importantly, we are using this sandwich type of ELISA. And it is a very sensitive ELISA, where first of all, what we will do, first we will put the antibody which is specific to any pathogen. For example, we have a virus here that is called apple mosaic virus or apple necrotic mosaic virus. So what we do in this DAS ELISA or double antibody sandwich ELISA, we will put the antibody that is called the primary antibody, and which is specific to that virus. So first we will put in this titer palette, micro titer palette. Then we will put the antigen. Antigen means the sap from this plant which is infected, which we suspect that this plant is infected with this virus. So we are putting the sap, and in the sap the virus is as antigen, and we will put that antigen. And then coming to the third step, that is the secondary antibody. In secondary antibody, we will be having one enzyme is there linked, so that When we put the substrate on that enzyme will react to that substrate and it will produce the color. But if this enzyme or this secondary antibody is not there, and enzyme will not be there, this substrate will not work and color will not be there. So if primary antibody is there, then antigen is there and the secondary antibody is there. And secondary antibody will react with this this antigen. And when this antigen will be there, only secondary antibody will be there, enzyme will be there, and substrate, when we will put the substrate, it will produce the color. So when color is produced, then we will say that yes, this virus is present. If this antibody, this color is not present, if color is not present, for example, we can see here, if there is no color, we can say, no, this is not, this virus is not present. So then another step will be there. For example, we can also relatively quantify the virus. For example, we will get the OD values or we will get the intense color. We can say the titer of the virus might be more in this type of a sample as compared to others. So this we measure this then on this ELISA reader, and we will go will detect that. I will quantify that virus through OD values, and we can come to know that what actually the virus titer is present in the sample. But there is one drawback, or I can say the demerit of this ELISA, that it is a almost 2-3 days process. First, then second, it is less sensitive. Less sensitive means, for example, I will give you an example. If we have a 10 particles of the virus in the sample, it will detect it. But if it will go down 8, 7, 6, it will not detect the virus. So the sensitivity level of this technique is very less as compared to PCR. PCR is, this ELISA is based on the proteinaceous nature, where we can use the protein. But through PCR, it is a molecular approach where we are using the DNA from the pathogen, or we are using the RNA from the pathogen. So for example, we have any this disease symptomatic plant, we will isolate the DNA, or we are having the fungus or bacteria, which we don't know the what is the name of this bacteria, what is the strain of this bacteria. So what we will do, we'll go for the DNA isolation or RNA isolation in case of viruses. Then after DNA isolation, we will go for the gene-specific amplification. Then we will go for the gene-specific amplification through conserved genes. Or even, for example, we have some conserved genes in the fungus like ITS, beta-tubulin, and elongation factors, which are conserved in the fungus. So these will, they have not changed throughout the evolution. So they will give the better results. But we can have sometimes the specific genes, especially in case of viruses, we are taking the code protein gene, or we're taking this replicase gene, we're taking the movement protein gene. Sometimes we'll be having those specific genes also by which we can detect the pathogen. So for example, in case of fungus, we will take ITS, we will take beta-tubulin, we will take elongation factor, and through this PCR, we will amplify that gene. And after amplification, what we will do, we can go for the sequencing. And after sequencing, there are n number of bioinformatic tools through which we can identify that pathogen. For example, we have taken this sequence and we have blast this sequence in NCBI. After the NCBI blast, you can see that the pathogen which we were targeting, or the virus which we are targeting, was apple stem eating virus. So through this PCR or through this sequencing approach, we can identify the virus. But here we should know at least first the primer of the pathogen or primer of the gene. Unless and until we don't have the primer, we will not go for the detection. Then another approach will be, it will be a same through primer only. But here what we will do, we will be detecting more than 2 pathogens in one group. For example, here you can see if we have to detect this apple necrotic mosaic virus, apple mosaic virus, or stem tipping virus, or stem green virus. Singly, we have to detect those through single PCR. For example, if it takes 20 microliter, 20, it will be 60 microliter, and in 60 microliter, there will be time, will be there, more time it will take, and more resources it will take. But if all those 4 will be detected in single PCR, in a single tube, in 20 microliter reaction, we will save the resources, we will save the time. So this multiplex PCR we are using for detecting number of pathogens. It is not only we can detect the viruses, but we can detect virus, fungus, bacteria in a single tube, so that we can have this multiplexing technique, which saves our time, which saves our resources as well. Then very important is that the real-time PCR. Real-time PCR is having various these benefits. One, it is very much sensitive. Sensitive, for example, we have a PCR, it is up to carry for -2, -12. But if we will see the sensitivity nature of this real-time PCR, it will go up to carries for -18, -17, -16. So if the titer of any pathogen, for example, these viruses, is very low, then only this real-time PCR will work. So you might be knowing that sometimes when the COVID problem happened in 2020-21, so some were asking that whether it is a RAT test or it is a PCR-based test. So this PCR-based test is more sensitive as compared to that RAT. RAT was rapid antigen test was based on the ELISA this. So that was not much sensitive as compared to this real-time PCR. So this PCR is very much sensitive. And one more thing, we don't need any this gel electrophoresis system. Here you can see the curves. So here we can come to know that yes, the curve has gone up. So this the pathogen or whatever gene we are amplifying, that it is present here. And through this approach, we can also quantify the pathogen, that how much the pathogen titer is present. And we can have also the absolute quantification, not only relative quantification, but absolute quantification using this real-time PCR. These are the approaches which we are normally using, but now some recent approaches have come, that isothermal this amplification. For example, in PCR or real-time PCR, we need huge equipment which costs us almost 3 lakh, 4 lakh. Even real-time PCR costs us 10, 11, 20, up to 15 lakh. So we need those things for those techniques. But in isothermal amplification, we don't need such kind of huge equipment for carrying out the detection or carrying out the amplification of the pathogen. This is the isothermal amplification, or we are calling this point of care based detection technique. In isothermal amplification, some constant temperature is needed. For example, at 65 degree or 50 degree or 30 minutes, the pathogen will this get amplified and we will detect the pathogen. And very important is their lab and loop mediated isothermal amplification. Here what we will do, we will design the primers. After designing the primers, almost 6 primers we need, and we are designing from any pathogen. Then from that pathogen, then we will design afterwards, we can use those 6 primers for the pathogen, which we will, for example, from anywhere come test to know which type of pathogen is. So 6 primers are needed in this lab, and it will amplify at almost 50 to 60 degrees Celsius in 30 to 35 minutes. So only we need small water bath, or even sometimes water bath is not required when we have standardized the things. Then we can use our body heat as well. For example, if we have standardized it at 37, 36 or 35 degrees Celsius, any pathogen, we can put in our body temperature also, and put for 10 minutes, 20 minutes or 30 minutes, we can amplify the pathogen. And sometimes you can see the turbidity will be there. Here I can show you. The turbidity will be there, or we are putting some calorimetric dyes, and when dye will be there, here you can see the coloration will give you the indication that any pathogen is present or not. If it will not give any kind of color, you can see no, this pathogen is not present. So this is a calorimetric based. Within 1 hour, you can do this pathogen test through lab. Then similarly, we are having now the recombinase polymerase amplification, and now we are using this recombinase polymerase amplification on small strips, just like we were using the RAT, rapid antigen test. So here we are using, but that we are in RAT, we were using that protein-based nature. But here we are using the DNA approach, and we are using the molecular approach, that is the DNA or the RNA. We are putting the sample DNA or RNA here. We have 2 lines, test line and control line. And for example, this is a test line where we should get, if sample is positive, we should get the color. And control line, if it is a positive, we should always get positive. But test line, if it will show negative, and control line will show positive, that means the pathogen is not present. If test line shows the positive color and control line doesn't show the positive color. That means our test is wrong. Control line should always give the positive result because it's a control. It's a control pathogen. It's a control virus. It's a control bacteria. So we have put already, for example, I will take the example of APNMV. We have put the APNMV nucleic acid there. So it should give the color when we will put the sample of this APNMV. And test line should also, if test line will give, then it is a positive. If test line will not give, it is a negative. And if test line will give, but control line will not give, then we can say it is a wrong test. So this RP is being now frequently used. Now we have are using this lateral flow assay of this. This is called the lateral flow assay, where we can detect the pathogen in 15-20 minutes. These are recent advances which through which these pathogens we can detect. Then the most important approach is the NGS, that is called the next generation sequencing approaches or high throughput sequencing approaches. In this approach, for example, mostly we are using these approaches in the viruses. For example, we are getting a type of symptom which we doesn't know that what actually this symptom has been caused by. So what we will do, for example, we will take the sample. I can show you here. So we were having the three these plants. One was the asymptomatic plant, which does not show any kind of a symptom. Then we were having the two symptomatic plants. One was showing the mosaic and necrotic type of symptom, and another was showing only the mosaic type of symptom. So we were thinking that it is the apple mosaic virus which is causing this disease. So we were just going like that only. But through this approach, this next generation sequencing or RNA sequencing, mostly which we are using in this approach, what we did, we went for the next generation sequencing or RNA sequencing of all these three samples. So to our observation, what we observed that in asymptomatic plants also what we got the three viruses, which otherwise we can say no, this is asymptomatic, no symptom is present. So there shouldn't be any kind of a pathogen. But what we find that this chlorotic spot virus, apple stem eating virus, we could also observe in that asymptomatic plant as well. So, for example, in case of these perennial crops, we need to be cautious about these viruses. So these viruses should not spread to the farmer's field. Maybe just like COVID, what the havoc it has caused. Maybe these viruses can also cause such type of havoc, and that is why the Clean Plant Program of India has came, so that we should give the plants virus free to the farmers. So then the symptomatic plant, obviously, what we are thinking that this is apple mosaic virus, but to our observation, what we see, apple necrotic mosaic virus. The new virus was observed, which is causing this necrotic type of a symptom. And in another wire, we thinking again the apple mosaic virus only, but we can again observe here the apple necrotic mosaic virus and apple mosaic virus was observed from other than these apple stem growing and apple stem eating viruses. These are mostly the latent viruses which does not produce any kind of the symptom. So these high throughput sequencing technologies nowadays mostly being used, especially exploring the virome of the fruit crops, because the perennial nature of the fruit crops is very important, and we need to have these plants virus free, because we are taking this sign wood, budwood from these plants, so that at least we should know that what kind of a virus spectrum is there, and that should not spread to the farmer's field. Thank you very much. These are some of the advances or some detection techniques which I have discussed. And even there are n number of techniques also. Now we are using the information technology. We are using this IoT based technologies. Now we are using by which we can take the images through drones and other satellite images. Then we can share those pictures. And now the things are changing. And now we are going to the digital world, and through artificial intelligence we can detect these pathogens. And also the sensors are very important, even biosensors. And we have developed one biosensor for the early scab detection, and that is totally based on the volatile. So that is now another recent advance, that sensors, biosensors are being utilized for detection and diagnosis of these diseases. Thank you very much. Any kind of question, I will give you. Thank you.
Hello.
Hello.
Thank you, sir. Sir, in chat box, there might be some question. Can you please go through the chat box?
Okay. Yeah, for example, down and up regulation, we can check only after going through the transcriptome analysis, and then we can, for example, they will give you one scale that this gene has been upregulated and downregulated. Then through real-time PCR, we can design the primers for those genes, and we will see this level of graph that how it goes, whether it goes towards the minus side or towards the this positive side. And based on that, we can check the upregulation and downregulation of the gene in the quantitative PCR. So if there is no question, we have actually one meeting at 11:30. That is why I was a little fast today. And thank you very much. Thank you, organizers, for inviting me for this lecture. And I hope you have got something from my lecture. And obviously, it is a short time. We can't explain each and everything and each detection technique, the principle, and how it works. So hopefully, in next training courses, maybe sometimes we'll discuss the detection techniques in detail. Thank you very much. Thank you very much.